Good afternoon, and welcome to the Second American Museum of Natural History web panel discussion on the COVID-19 pandemic. The discussion today is on vaccines, testing, and the science behind a cure. My name is Rob DeSalle, and I'm a curator here at the museum in the Institute for Comparative Genomics. Before starting, I'd like to thank Ellen Futter, president of the museum, Mike Novacek, provost of the museum, and Lisa Guggenheim, senior vice president of the education department, for their strong and rapid support for these events. And a big thanks goes to Bella Desai for her amazing work in organizing these two events. We're excited about this second panel discussion to educate the public here in New York City and across the nation about the pandemic. While our doors have been closed to visitors for several long months now due to the pandemic, we feel we have a responsibility to fill the information gap that has arisen recently and to be as helpful as possible in giving the public the necessary information to navigate the next critical months. The AMNH has twin directives in education and science. You can visit our webpage, www.amnh.org, to view the ways our scientists and educators have responded to the pandemic. Our scientists here are making valuable contributions to the global scientific fight against this pandemic. To complement the research we do, over the past 2 decades, a dozen exhibitions have been mounted focused on public health and infectious disease at the AMNH. These include Epidemic, Countdown to Zero, and Inside You. The museum has also been involved in citizen science concerning public health with our collaborative effort with Columbia University called the Healthy Human Microbiome Project, an exciting scientific study conducted with our visitors to find out more about the microbes that make and keep us all healthy. Again, today, we extend this commitment to public health information to the current pandemic in the hopes that knowledge of the science of this disease and the virus that causes it will give you answers and advance us closer to the end of this pandemic. Today's event will be moderated by Arielle Duhaime-Ross. Arielle hosts the popular podcast Reset, a technology-focused broadcast produced by the Stitcher and Vox Media Podcast Network. Her interest in science is broad and covers topics that include environmental, human health, and LGBTQ topics. Arielle's most impressive work concerns examination of ethical issues that are ubiquitous in scientific application to human health. She is a former reporter for the Vice News Tonight and earned the Herb Lampert Science and Society Award while reporting for Vox's The Verge. Arielle is also an accomplished writer with articles in Scientific American, Nature Medicine, The Atlantic, and Quartz. Her latest work is on COVID-19 long haulers, a fascinating story I hope you get a chance to look at. I've been privy to the discussion about today's topics and the questions Arielle intends to pose to our panel and can't wait to get the panel rolling. So now, Arielle, I will hand things over to you.
Thank you so much, Rob. Yes, as Rob mentioned, my name is Arielle Duhaime-Ross. My pronouns are she and her, and I'm really excited to be moderating this panel today. I won't say much more about myself. I'm here now to introduce our panelists. And they're great panelists, by the way. So our first speaker is Dr. Pardis Sabeti. She is a computational geneticist at Harvard University and the Broad Institute, who's been working on creating diagnostic tests for COVID-19.
Hi.
There's Dr. Sabeti. Our second speaker is Dr. Kizzmekia Corbett, a viral immunologist and lead scientist on the vaccine development effort at the National Institute of Health, at the Vaccine Research Center there. And our third speaker, sorry, go ahead.
Hi, everyone.
And our third speaker is Dr. Lynne Richardson. She is an emergency physician and the co-director of the Mount Sinai Institute for Health Equity Research. And I'm not gonna spend too much time on this. We have a limited amount of time, so I'm gonna dive right in. Our first question is very broad, and I think it's going to be a good way to sort of ground this discussion, which is just, what exactly is a virus?
I guess I'll tackle that question. It is a pleasure to be here today, and a little bit of a breath of fresh air to talk about some of the more broad topics surrounding this pandemic and vaccine development, etc. So a virus, it is a very small, what we call microorganism. So you can't see it with the eye. It is a organism that evolves to infect different species. May have heard that there are coronaviruses, for example, that infect animal species like bats. There are also viruses that we're well aware of that infect humans. And the way that these viruses do that is because they have these molecules on their surface that attach to cells in whatever species, whether it be a human cell or a bat cell. And then by way of attaching to that cell, the virus then drops off its genetic material inside of the cell. And then this process, such as replication, starts to occur. And then the virus makes daughter viruses or little baby viruses. And then you have what you would consider to be a viremic or an infection. And so those infections cause manifestations, like, for example, with the coronaviruses, lung manifestations, so coughs, sneezes, severe lung damage in some cases, etc. And so one of the things that they wanted me to touch on was how viruses evolve, because as we have become aware through this coronavirus, is that one day it was not in a human being, and then the next day it seems like it was. And one of the ways that that happens is that first interaction of a virus with a cell applies pressure to the virus. So even though a virus doesn't have a brain, it is constantly, quote unquote, thinking about a way to get into its new host and cause disease and then be able to spread all over the world like this coronavirus has done. And the way that it does that is by making mutations in the part of the virus that binds to the cell. And so over time, those mutations accumulate and the virus becomes more adapted to the particular cell type. And this happens not just in coronaviruses, but it happens across a wide spectrum of viruses, HIV, influenza, etc.
All right. That was perfect. I am curious, though. I think that prior to January, December, for some people, the term coronavirus was not necessarily super familiar for a lot of people. So in the grand scheme of things, where does this particular coronavirus fall? Like, are there a ton of different types of viruses? Like, how should we be thinking about this particular coronavirus?
You should be thinking about it how you think about your family and your cousins. So if I am coronavirus A, or in this case, SARS coronavirus 2, then I have a closely related cousin that looks a little like me, about 80% related to me, and that's SARS coronavirus 1. And then I have some distant cousins that also can infect humans. There's a total of now 7 of those that can infect humans that are a little bit distantly related to me, and they, between 60 and 70% related to me. And then I also have, hundreds of other cousins that can't infect humans yet. Maybe have the potential to one day evolve to infect humans, but those are zoonotic or animal coronaviruses that are even further related to me. So if you think about coronaviruses or any viruses, the way that you think about your family and how related you are to members in your family, I think it's a really easy way to grasp the concept of viral families.
So what I'm getting from that is that there's actually a very broad diversity of viruses.
There are a broad diversity of viruses in general, and then obviously within each family. So there's about 24 different viral families that we consider to be dangerous or potentially dangerous or pandemic potential for humans. And so in each of those families, there's, hundreds of different cousins in those families.
Okay. All right. Okay. So the reason why I wanted to talk about that is because I can only assume that has an impact on this whole idea of a vaccine, right, and vaccine development. But we're going to get to that a little bit later. But I want people to keep that in the back of their minds as they listen to this conversation. So along the same lines, right now, I'm not talking months ago, I'm talking right now. What do we know about transmission of this particular coronavirus? Like, how should we be talking about that?
Someone else want to take this one?
Yeah, does anybody else want to jump in?
I'm happy to, but I think
sure.
There's immunologists in the group, so I feel like we want to hog the show. But yeah, so what we know about transmission of this particular coronavirus. So what we know is that it's a respiratory virus. So it is a virus that lives in the lungs, And particularly for this one of the reasons why it transmits so well is because it does a very good job of replicating in the upper airway. So right here where you snot, and where, if you sneeze, there might be some droplets that might touch another person and be able to infect another person. And that's actually how it is transmitted. It is transmitted through what we call droplets. So those can be microscopic, and they transmit from person to person through sneezes, even, it's been said talking or just being face to face, very close to one another.
Okay. I'm sort of wondering, Dr. Richardson, yes, I actually was going to ask you my next question, so please go ahead.
Okay, yeah, I just want to add to that as a clinician. So as with most viruses, or most of the respiratory viruses, and coronavirus is not the only family of viruses that cause respiratory disease. There are actually many of them. Most people have heard of influenza, and there are others, RSV and so on. But in addition to the sort of direct spread of droplets, because your face is close to the face of someone who is, who has the virus, who is shedding it, it's also possible for the droplets to land on surfaces. And then you touch the surface, and as most of us are doing almost constantly through the day, then we put our hands near our face. And so it's not just sort of face-to-face transmission, it's also face-to-hand and then hand-to-face transmission, which is why washing your hands is such an important interruption of that transmission chain. And so it's not enough to just sort of keep your face distant, but you also have to be very careful about washing your hands frequently and trying to keep your hands away from your face.
Okay, so with that in mind, I would love for us to talk a little bit about what are our best tools against this particular virus, right? Right now, forget about the vaccine. We can talk about that later. Right now, what are our best tools to fight this current pandemic? And Dr. Sabeti, I'd love to hear from you.
Sure, yeah. So, I think that, one of the best tools is detection, is being able to just identify when, I think actually when I think about tools, I think about education and community is actually probably our most important tool. Our most important tool is to be educated and to work collaboratively. If we actually work as a community and support each other in the right ways, and really think about how to support each other and how to exist as human beings, that's the number one tool in our arsenal. Happy to talk about that more, because I think about that a lot. Then, from there, it's detection. It's having diagnostic tests available as far-reaching as possible to give as many people as much agency on the front lines as possible to be able to pick up cases the second they occur, and to then be able to, track down those cases and stop further infection spread. And then, of course, there's a lot of therapies and vaccines that are in development that are going to be critical and that are coming down the line. And there's already some promising results from therapies that were already available, that like remdesivir, which is something used for other viruses that could now be useful here. We're taking things that we already have at our fingertips and trying to see if it might work here. And then we're also trying to find other approaches to either treat it or to prevent it through things like vaccines. Those are the different kinds of tools that we have. And any one of them, like I said, I'm happy to go into much, much further.
Right. So there's behavior, and then there are sort of the medical approaches. There's the testing. There are the medicines that you can give to somebody who's already infected. And then there's hopefully vaccines down the line, or immunity in some way, shape, or form, right?
Yeah.
So let's talk about testing a little bit more. What are the different tests that are available to us? And I would also love for us to sort of address the whole issue of false negatives, false positives. How should people be thinking about the tests that are available to them right now?
Sure. I can take that. So there are different kinds of tests. There's a lot of different kinds of tests. So we have a matrix that we always do. We're like, okay, there's many different ways of testing. But what I would say is there are, as far as what you're trying to capture, the key things is you're trying to capture either that you have the virus, like you're carrying the virus right now. And you can do that by either trying to, the virus has proteins, and you can try to capture to find something that we can use actually antibodies that can capture the proteins of the virus, and that's one way. It's called antigen capture, but it's a way of basically trying to detect that there's the virus and you can and see it. The virus also has something called a genome sequence. So every organism on Earth, including viruses that are sort of in this semi-lifelike state, have a genome sequence. It's a string of letters. We call that DNA or RNA, if you've heard of it. And those are almost like barcodes that tell you what's circulating. If you detect a string of sequence that is associated only with SARS-CoV-2, then you can say, oh, you have that. And so there are a number of molecular tests, and the kind of commonly used one now is this RT-PCR test that is picking up the virus's genome. So that's another class of tests. But both of those are essentially trying to find out, are you carrying the virus in some fashion right now? And then on the other side, there's something called serology that we talk a lot about. And those are the kinds of tests that are picking up your immune response. So they're detecting your host immune response to it. That usually happens not immediately. So it's actually better at capturing it a few days after you've mounted an immune response, but then also lasts a longer time. So it's a way, a lot of people are talking about it now as a way of surveying communities and saying how much of COVID was circulating, how much SARS-CoV-2 infections do we likely see? And so those are the different kinds of things that you'll see. And then, so that's sort of one grid of what are the kinds of things you're trying to detect. And on the other grid, it's sort of like, do you want it to be point of care? Do you want something that you could do at home? And there are all these people trying to develop spit tests that can happen on lateral flow, like a pregnancy test that you could do at home, to massively high throughput tests where you could do thousands of tests at the same time, to other versions where you could actually test not just for SARS-CoV-2, but something our group is developing are ways of doing high throughput, but also testing for lots of different viruses at the same time. Because we know that a very small number of individuals that are suspected to have SARS-CoV-2 I'll actually have it. And then you leave and you're like, oh, but I have something, but what do I have? I don't even know. But I have a sore throat. But so can we give you some other answers? And things that circulate with at the same time are flu A, flu B, RSV, enteroviruses, all sorts of other things. And so can we actually test for all the possible things that might make you infected instead of just giving you a yes or no answer on one thing?
All right, that's really helpful. I am wondering though, Dr. Richardson, you are an emergency physician. How are you talking to patients right now about the idea of getting tested? What are you telling them?
Well, actually, it's interesting because a lot of patients are actually coming into the emergency department telling me, doctor, I need the COVID test because my supervisor says I can't go back to work until I have a test. So there are a lot of aspects of society that have latched on to this idea of testing, and they seem to be making up rules, often not really that are congruent with the science about how testing works. So I think when someone comes into the emergency department, if they have symptoms that are consistent with COVID, then obviously we test them as part of the diagnostic workup. As was said previously, it is possible that they have some other viral illness. You know, we've been dealing with respiratory viral illnesses for a long time before COVID was on the scene. And like COVID, most of them are self-limited. Most people get sick for a certain period of time, and most people recover. That is also true of COVID-19, although because it is brand new to the population, because no human had any kind of immunity to this virus, and there was no vaccine, the sheer numbers of people who have contracted it have been really unprecedented. But it is not in its behavior so very different from other viruses that we've had. The reason that it is a pandemic is because it is new or novel. And this is a scenario that clinicians and public health experts have talked about and planned for a long time. So, you know, for me now, testing is in large part a public health issue. It's important for us to know how much penetration there was. We know, for example, that you may have contracted COVID and not had symptoms, but you now have antibodies. So you had what we call a subclinical case. Some people did not seek medical care. Indeed, during the peak of COVID in New York City, we were actively messaging that if you were a little sick, you should stay home and you should only seek medical care if you were very sick, in an effort to keep the healthcare system from being overwhelmed. So it is, I think it's important for us to understand the penetration of this into communities. Even today, it does not affect that much my initial treatment. And in fact, in the emergency department, if you come with respiratory symptoms, even if we do a COVID test, it's likely that we will not have the results of that before the emergency department visit is complete. My decision about whether you need to stay in the hospital or you can go home and self-isolate really depends on how sick you are, not the results of your test. Now, the idea of having a test where we could identify whether or not you have COVID before you get sick is very attractive, because that's a period when a lot of the spread happens. I think at this point, most people understand if you're feeling sick, you really stay home, distance yourself from others, wear a mask. But there's a period where you've been infected and you are spreading the virus, but you don't have any symptoms yet. So the idea of having a test that could identify it very early is very appealing. And I think the other thing that would really be an important advance is a treatment that decreases viral shedding. In other words, it decreases the transmission of the virus from one person to another if it's taken early in the course of the disease. But as was pointed out, this is all brand new. When this epidemic started, there were no tests. Now there are lots of tests of varying accuracy. You know, you asked about false positive, false negative. That is true of any test. The newer a test is, the more likely it is that it's not as accurate and reliable as a test with which we have more experience. So all of those factors are in play.
Okay, all right. I am curious. I think, Dr. Corbett, you mentioned mutations earlier, and you know, viruses mutate. That's just a thing that they do. It's totally normal. I would love for Dr. Sabeti and Dr. Corbett to talk about mutations with regards to testing and vaccines. Like, is that something that scientists are thinking about and maybe even worrying about?
You want to start, or should I start?
Yeah, I think I'll start and just talk about mutations, and then I think you can really address how they relate to the vaccines. But yeah, so viruses indeed mutate. All genomes over time are mutating every time they're replicating. It's just that viruses are replicating much, much faster, and they're making lots and lots of copies. So there's a lot of opportunities for mutations to occur. And they also have RNA genomes, which is a kind of genome that mutates a little bit faster. That said, the specific, if you know sort of the specific machinery that replicates this particular SARS-CoV-2 virus is a little bit better than others. So in the grand scheme of things, it's not quite as fast as a Lassa virus or an HIV, but it's much faster than we are, for example. And so, but then compound on top of that it now had millions and millions of chances to replicate across lots of different people, and you're going to see mutations. And I think at the last count, there are over 300 mutations that have popped up and gotten to an appreciable frequency, like over 1%. But basically at this point, every position in that genome is probably mutated at least once. And over 300 new changes in this genome have emerged. And any one of them could have some biological impact. The vast majority of things just get thrown away. It's just nature's way of just trying to try things out, and most of them just get thrown away. But the problem is if you give a virus so many opportunities to spread, just that lightning in a bottle might happen, right? Like lightning might strike. And there's evidence now, there's been evidence in every outbreak that we've been really worried about, there's been evidence of a mutation that's occurred that made us worry about it. During the Ebola outbreak, there was a certain point where it was like, the trajectory was bad. It was already bad, and then suddenly it was like very, very bad. It just shot off, and you remember it's like under the people are talking about it, but then suddenly you're like it's everywhere. And at that same time, a mutation seemed to have popped up in the virus's glycoprotein, where we and others have shown evidence that increased the infectivity of the virus. And we just published a paper with Jeremy Lubin, my partner in crime and the person who leads all this work. And we and others have been publishing papers showing that there is a mutation in SARS-CoV-2 spike protein. It's called D614G. You may be hearing about it, but that also appears to be increasing the infectivity of this virus quite appreciably. And so, this is all early evidence, but it's something we shouldn't be surprised about. It's a reason why we talk about you don't want to rest on your laurels, because this is a moving target. It's a very volatile situation. And, even when people are like, your response shouldn't be, it affects like older people. It's not my problem, because hopefully you have family that you want to respect and take care of. But even if not, like it's also can change. It can affect you too. And so don't be complacent in any of this. And those things can affect diagnostics. So they can affect the biology of the virus, but they can also affect diagnostics. And if they work, if the virus changes against the targets, vaccines and therapies. And I think Dr. Corbett can get more into the specifics of the importance of vaccines.
I just want to flag one thing. I think probably a lot of people will be wondering about the word strain, like a different strain. We may be familiar with the movie Outbreak. Like, at what point is it okay to use that word? And at what point should people be freaking out by what you just said, Dr. Sabeti? Or should they be like, oh, it's just a natural process and this happens all the time?
I would say, so the kind of term of when we say it's a different strain, it has changes, and there's certain points where it's like, don't get into there's like a whole group of people who are like cladistas who like fight over like when is it new, when is it not. So I won't get into that territory, but yeah, it is changing. And at certain points, we might say this is a new version that we're talking about. It is neither like run out and freak out, nor is it rest on your laurels. It's exactly that. It's that it's not like, it's sort of when people talk about this idea of mutation, it's sort of, when we compare like during Ebola, we're like, when will it jump to go airborne? It's not usually like a one-click process, but it's a series of processes. The more chances you get for it to spread, the more chance those series of processes. So we saw in Ebola, it was like the glycoprotein changed and it became more infective, and then multiple more mutations happened on that background. So it just keeps picking up that ability to take speed. We see it also in malaria resistance to anti-malarial drugs, that the first one took 16 years to develop. Now it takes 6 months. Somehow, over time, these things do, I wouldn't say learn, but they are moving in a direction that makes even more changes more possible. So don't freak out. It's not suddenly changed, but do take it seriously and work to try to stop it.
All right, and Dr. Corbett?
Yeah, so I just kind of want to highlight something that Dr. Sabeti said that is interesting, in that this happens. It's almost the virus's job, so to speak, to mutate. So we do have to take, that knowledge as our backdrop and our understanding around vaccine development, and we do. So part of the reason why I've been working with coronaviruses is because when I started my current position, it was in the offset of the MERS epidemic or outbreak. And so I started working on MERS just out of curiosity and interest. And a lot of our research with MERS has really guided the vaccine development that we are doing now. A lot of those findings, for example, particularly around different mutations that have evolved in the backbone of MERS, which is another coronavirus that have evolved in the backbone of MERS, is that the type of vaccine that we are developing and that many people are developing creates an immunity that is broad in the sense that it encompasses antibodies that are polyclonal, so a lot of different shapes and forms, colors of antibodies, so to speak, that will cloud the spike protein in so many different directions and ways that one particular mutation in the spike protein, unless it is a mutation that is an escape won't necessarily have any effect on vaccines in the intermediate. People have modeled the mutation rate for SARS-CoV-2, and again, like she said, it's a slight mutation rate when you think about viruses, because coronavirus is inherently proofreading mechanisms. And so people have modeled it, and vaccines, if they create the type of immunity that we think they will, should be good in the upcoming several, 3 to 4 years. And so I think that we have to just understand that mutations happen. We have to, as vaccine developers particularly, make sure that we continue to scan the literature and look for them, and to understand where they are on the particular vaccine design constructs and how it might affect the immunogenicity. But I don't think we should be worried right now about some mutation popping up that's going to evade vaccine immunity for any vaccine that might be available next spring.
Great. That's a great answer. I mean, basically what you're saying is your scientists that are working on this are future-proofing this vaccine with the knowledge that viruses just mutate, and that happens.
Right. And I think that people kind of, as the field explodes with scientists, no longer just my home turf, I think that people are thinking about not just vaccines for today and, the current virus, but vaccines across the family. And, I think that people are thinking with the understanding that, number one, this particular virus will continue to mutate, albeit slowly. Particular viruses in the coronavirus family might evolve to infect humans again and cause this type of pandemic uproar. So we're thinking about it. And right now, there's no real cause for concern that any of the viruses, SARS-CoV-2 viruses, will evade the type of immunity that we plan to elicit with the vaccine.
All right. Now that we're fully in the topic of vaccine, I just want to get a little bit more on sort of the mode of how the vaccines that we're thinking of developing right now, the vaccines that we're currently working on, how exactly are they working in the body, right? Like, what is the thought process here? Are they different from other vaccines that we typically develop for viruses? How should what do people need to know about that?
So there's several vaccines in just the global pipeline for COVID-19 vaccine development. Generally speaking, they all encompass a part of or the entirety of the spike protein, which is the protein that's on the surface of the cell. Is that protein that I initially talked about that's responsible for attaching to a cell and causing the virus to get into the side of the cell. And so the purpose of a vaccine would be to, in so many ways, block that interaction or some part of that interaction, and then stop infection, or at least impede infection and replication. And so people are placing the spike protein in various modalities. Ours particularly is a messenger RNA modality, but you can give just the spike protein to a person and it can create some level of immunity to it. You can give an activated virus that has a spike protein on the surface and create immunity to it. So there's various modalities to get an immune response to that spike protein. And so they all will probably work in the same way, and that is by eliciting antibodies that will be protective against the virus later.
All right. Now I guess
I have to say this because I'm a
go ahead.
girl, and then someone's going to be a T-cell girl on the other end and scream at me later. So there are obviously other immune mechanisms that are influential in protective immunity. Other cells, like T cells, for example, help as well.
All right. Okay. I'm glad you put that in there. Okay. So I feel like now is the time for the important question that is on everybody's minds, which is how soon before we actually have a vaccine? At the very beginning of this pandemic, we heard the 2-year figure quite a bit. I know that figure refers often to development and not manufacturing of the vaccine. So I really want us to tease this apart. How soon before, like, somebody living in New York City or South Carolina can walk into a clinic and get this vaccine?
I think that so the 2-year in the beginning was not my quote. I'm sure of that, because part of the entire job that I have under the umbrella that I have at the Vaccine Research Center is around pandemic preparedness. And so we wanted to initially start it as a prove a point, but now it's a problem. So this is what we're going to do. We said that we would try to have a vaccine developed by 16 to 18 months after the onset of the sequence. And I think that the trajectory remains the same. So we're currently in phase 2. Other vaccine products are also in phase 2. I think that there haven't really been any signals with any of the products that has suggested that the trajectory of the phase 3 trials should cease. So I think you should see vaccines going into phase 3 in upcoming weeks or months. And I think that if all goes well, next spring.
All right. And when we say next spring, you know, this next spring thing, is that like, oh, we have a vaccine that works and we know how to make it, right? That's what you're referring to?
So I think, overarchingly, companies have already pulled the trigger on manufacturing for their vaccine products, particularly if they have promising clinical trial data, because the amount of doses that it's going to take to vaccinate even the United States, let alone the world, is robust. So I think that start a vaccine in general population with an FDA approved and licensed product by next spring.
Okay. All right.
Yeah, if I could just say,
you know, it's really important. I think the point that you're making, Arielle, that it's one thing to have a proven, ready to go, FDA approved vaccine. It's another thing to have, 200 million doses of it available to immunize people. And so there is this tension. And one of the things that people who talk about, responding to pandemics talk about a lot is the need to actually start large scale manufacture before you've completed dotting all of the I's and all of the T's on all of the tests. And to do that with multiple different vaccines, because you don't really know halfway through which one is going to prove to be most effective. Now, on one hand, that may seem very wasteful, because this means that you will wind up developing millions of doses of a vaccine that you will then throw away when it turns out this other company's vaccine is better, and that's the one that we should go with. But when you think about the economic costs of doing the social distancing necessary when we don't have a vaccine, that becomes a trivial investment to really mass produce multiple promising vaccines. Now, it's hard to see that that's actually happening in a coordinated way at this point in time. And there is a danger because a company does make an investment in producing a vaccine that's not yet FDA approved, but perhaps is available for compassionate use or an emergency use authorization. And so we really have to keep a close eye on this. And I think that we as public health experts, as well as clinicians, to make sure that the vaccine that gets to the public is known to be safe and effective, and that while we do need to mass produce vaccines that ultimately won't be the one, we need to make sure that, they don't somehow find their way into large numbers of individuals just because they were mass produced. So there is this tension here.
Yeah, I think that, my opinion is that, I think that that's the FDA's task. That's why we put together these large and very stressful data reports. But I think, I think that one of the things that, that you said that really stuck with me is that sometimes it's wasteful, right? With all these companies start pulling the trigger on their manufacturing, right now, et cetera, perhaps it will be wasteful. And when you think about it from a business perspective, that's the reason why pandemic preparedness has largely gone so under the radar, is because we had the MERS construct inside of mRNA 3 years ago. But who's going to pay millions of dollars for a phase 1 clinical trial and manufacturing of the clinical grade product, et cetera, when the epidemic has largely waned with some steady state transmission only in the Middle East. And so we have to start thinking about how we, can start to do this earlier, because the truth of the matter is the phase 1 could have been a little bit shorter or phase 1s, multiple, not just ours, could have been shorter had there already been a vetted coronavirus vaccine in whatever particular modality, whether it be mRNA, et cetera. Right. But that takes money. This manufacturing that everyone is doing really out what is called at risk in the business world is taking money. But there is the weight of what happens if that is always kind of in the backdrop. And for sure, I haven't done the financial modeling because that's not my expertise, but I do know what it looks like, what pandemic preparedness looks like. And I do know that it's probably expensive. But I do think that it will be cheaper in the long run than any of the outcomes of this pandemic or any future pandemics.
Well, that's true of every aspect of emergency preparedness, right? Yeah. But nobody wants to spend a lot of money preparing for something that you hope will never happen. And then if you do that, everybody feels like, well, that was wasted money. And I wasn't saying that it's wasteful. This is what we should be doing. We should be mass producing all of the promising vaccines so that when we find out which one is the right one, we don't then have to start ramping up the manufacturing. My caution was about, we should make sure that those, those vaccines that didn't work as well, that they all get thrown away, not that they somehow get, pushed out under various pathways just because the company has now spent money making them. They're looking for some way to mitigate that, but that it's almost a whole other conversation. But I just want to say it doesn't just apply to vaccines. What we saw in the healthcare system, despite a lot of talk and a lot of planning and a little bit of preparation, was how unprepared much of our healthcare system was for the kind of surge that needs to be done. And it's not because no one had ever said, we're going to need, 2,000 extra hospital beds in New York City if there's a pandemic. But, when the governor and the mayor got the healthcare leadership together in early March and said, you need to create 2,000 additional beds in New York City. And the house like, what? How are we? It's like they had never heard the concept. Although I know that I have been, looking at and reading surge planning documents for 2 decades. So I hope one of the things that this pandemic will teach us is that we need to make an investment in preparedness. We need to do it on the vaccine development side. We need to do it on the healthcare system side. And that means that you have extra resources that are available to be rapidly used when you need them. And that means that they are not busy producing revenue and doing other things on the healthcare side. Right now in the US, hospitals have to operate at like 90, 95% capacity just to break even. Mm-hmm. That's the way the healthcare system is set up. So there is no slack resource. Nobody's invested in that. And maybe we need to as a society.
Yeah, I completely agree with that. I want to follow up with a question to you, Dr. Richardson, if you don't mind. So I kind of have written these commentaries on pandemic preparedness, but from like the vaccine development standpoint, with bullet points on what, like, if a government really wants to do it, what should they do? Mm-hmm. Can you just give me like the 3 bullet points, what that looks like from preparing from the healthcare side, or 5, however many bullet points you want?
Yeah, well, I mean, it really is. I mean, it's facilities and equipment, staffing, and coordination, right? So it's not rocket science, but it does take an investment. I was quite involved with the response of the Mount Sinai health system here in New York City. We're a 8 hospital system 7 acute care hospitals. And just the level of coordination that we were able to achieve as a healthcare system, I think made a tremendous amount of difference, as opposed to them being 7 separate hospitals, each trying to figure out how to deal with this. We were able to move around personnel. We were able to move around equipment. We were able to move patients around to smooth census. When Queens and Brooklyn, our smaller community hospitals, turned out to be the busiest in terms of the numbers of patients who were presenting very sick, we were actually able to move patients around the system to smooth that out. And in an ideal world, that would have been happening on a regional level and a national level. And so it requires advanced relationships and all of that coordination. We are a big country, and we're seeing now COVID did not peak in every portion of the US at the same time. We should have been able to move resources around, staff, equipment, medications, so that no place ever got overwhelmed. Because when we were, in trouble here in New York, they should have been, sending us ventilators from Florida. And now that Florida is on fire, we should be sending, but that national level coordination, unfortunately, is not happening. But it is possible to prepare for this so that the healthcare system in one place never gets overwhelmed. New York City did a magnificent job, I have to say. But you have to have the equipment, you have to have the ventilators, you have to have the PPE, the protective, the personal protective equipment to keep employees safe. There's certain kinds of staff you need. We ran, respiratory therapists were at a premium. When you have several hundred extra patients in your hospital on ventilators, you need respiratory therapists to manage them. You need just-in-time training so that people who usually do something else, which has been suspended because of the pandemic, now get trained to manage the vents. And so all of this was done actually pretty impressively, somewhat on the fly within the Mount Sinai health system, but we could have been much better prepared to do that because this was all predictable. So, I just listening to you folks talk has been really interesting because I used to be a climate change correspondent, and it is really hard to listen to you and not see the incredible parallels, right? Absolutely. We're talking about disaster preparedness. We're talking about how humans are terrible at preparing for disasters because we don't like to think about them and we don't want to spend the money because maybe it won't happen in your lifetime. And really, I think that if there should be some takeaway from all of this, I'm hoping that it's that actually we do need to be prepared. And Dr. Sabeti, I would let you talk, but I also want to just flag that we have some questions from the audience and we should probably move on to those, but please go ahead.
Well, no, I think the only thing I'll say is that I totally agree, but to kind of speaking to the cost and all of these other things, I just want to note that, well, one, infectious diseases have caused more deaths and more economic loss than all the modern wars and all the great wars, right? So on an annual basis, the flu causes more deaths than we see in most wars. So it is interesting because we have a massive defense budget, but we don't have a budget for this. We're preparing for something that we're hoping will never happen in the defense budget. So we're used to preparing for something we don't want to happen, but yet we aren't preparing for this. And kind of speaking to the climate change component, I still, infectious diseases are one of the only existential threats to humanity for which we have an annual exercise that would prepare us. If we actually could stop the flu going through the office and whatever Janice made you sick made me sick, we'll stop a raging pandemic, right? If we actually had better diagnostics and vaccines for MERS and SARS, we'd be better prepared for that. Like, we keep seeing these things over and over again. So it's both that it actually does cause more death and economic loss than the military. So we should spend at least, and we're not even spending a fraction what we spend on the military budget. And also it's an existential threat for which doing something now has an impact. It's not waiting for something. It's actually the detection you need to detect any virus is kind of the same. It's a little bit of plug and play. There's some subtleties, but if we had better vaccines for other viruses, we would have a better vaccine now for this. So anyway.
That actually leads us directly into our first question from the audience, from Nicole Davies, who asks, if viruses are related to each other, why can't we use a vaccine for a related virus against this particular coronavirus, right? Why can't we use that knowledge that we already have?
Well, okay, so because we don't have a vaccine for any coronavirus, even the ones that circulate every single year and cause, the sniffles around flu season. There's 4 of those. We don't have a vaccine for those. Largely, the vaccine development that had happened for SARS and MERS when those outbreaks occurred was halted, not because the vaccines weren't good enough, but just because the pandemic or the epidemic died down to a point where it was not worth going into clinical trial and really testing those vaccines in human. And furthermore, certainly not from a business perspective, licensing them for use. And so we know that there is some what's called cross-reactivity between different coronaviruses. We know that, for example, COVID-19 directed antibodies have been isolated from people who had SARS-1, 15 years ago, 12 years ago. So we know that there is some cross-reactivity, but the type of vaccine design that is necessary to really elicit the type of cross-reactivity that you need to cover an entire family is something that is going to take, a level of investigation. And so I think, there has been rhetoric. Why can't we just use the flu vaccine for the coronavirus? Because, we say, oh, flu-like symptoms. And so that rings true to people and it's completely understandable. But these vaccines, the way that they just the way that they work and the way that they trigger, so to speak, your immune response to see things is pretty specific, actually. Oftentimes to a specific quote unquote strain of a virus within a family.
All right. Thank you so much. We have another question. I think Dr. Sabeti might want to take this one from Rachel Fullreader, asking, when is universal testing going to become available?
Oh, wow. That is a good question. I think that's the $1 million question. It's hard to exactly say just because there are, I think, so many different diagnostic tests that are coming down in FDA, down the FDA line. And they're all kind of happening simultaneously. And I think that what we're going to see probably in July and August is a whole series of more FDA approvals for new diagnostic modalities and testing. The bigger question is actually manufacturing as well, because the issue is that, there are multiple companies that have really good diagnostic modalities already available, but it's just how to manufacture it fast enough. And it's such an interesting dynamic because those companies are also then themselves having to deal with social distancing. And so you're trying to work as fast as you can under an environment where you are really hamstrung. Kind of going off on a little bit of a soapbox, we haven't really thought about that either. Like if you really think about this kind of scenario, you would have a lot more lab facilities that would have a sleeping housing right next door, and you'd probably close them in and have people working around the clock, as opposed to having people commute in and have to worry about COVID coming in. But anyway, on a side note,
we actually thought about that.
Yeah.
We were like, should the team just live here?
Yeah.
It was a serious thought when we first shut down. Should we all live there and work? Obviously, that's not really ethical, and it didn't go down that way. But yes, it is something. No, seriously, if there was a structure in place for that kind of thing, where you could pull the trigger on that type of thing, I think it would work great in these types of instances.
A lot of hospital staff actually did that because they didn't want to take the risk of taking the virus home to their families. So we had physicians and nurses who were sleeping either in hospital provided housing or hotels for 2 months during the peak of the virus, so they wouldn't take it home.
Wow, that is a huge sacrifice that people are making, right? Not seeing their families, never leaving their workplace, which is the stressful place in the first place. That's a huge sacrifice.
Yeah.
We have another question from Cubs fan. Why is this virus more deadly than SARS?
Yeah, so it's not. It's not by far. If this virus was as deadly as SARS, what is happening right now with the numbers for the amount of deaths would probably be 5 or even 10-fold higher. So SARS had about a 12% case fatality rate. We're at a 1% case fatality rate with this particular SARS-CoV-2 virus. What you're seeing happen is not that it's more deadly than SARS, but if you compare red apple to green apple, so to speak, SARS being the green apple just wasn't really able to sustain its human-to-human transmission, largely because the viral infection was so low down in the airway that there wasn't really robust transmission of droplets, so to speak, from a person to person. But here, we're just seeing that there's robust, sustained human-to-human transmission. This virus binds to human cells, gets in there, makes viruses, and those viruses are being spread via the transmission mechanisms that we discussed before in a very, very efficient way. And so that's the real two differences between the viruses. I hope I don't ever see a pandemic on this scale with the type of case fatality rate that SARS had. That would be terrible.
All right. Okay. Our next question is sort of a two-parter. One is from the audience and one is a mix of things that I think we should address, which is just like the idea of herd immunity, right? I think that there is a perception that acquiring immunity naturally by means of actually just getting sick is a better way of getting immunity than through a vaccine. And I would also like us to talk about the vaccine itself, how much of the population needs to receive a vaccine, and I mean the worldwide population, before we can say this pandemic is over. Those are two very large questions. I would love for us to talk about that in the 3 minutes we have left.
I can address both of them very briefly. Firstly, the way that I think about herd immunity does not include people having to die, possibly. Okay? If the chance that you might die from the thing that's giving you herd immunity, I don't think that that's the way that we think about herd immunity. Herd immunity is a term that came from the field of vaccinology, whereby a percentage of the people in the population are vaccinated so that there is not sustained transmission of that particular virus within that population. Not that a percentage of the people go out and get coronavirus, possibly could die, and then are maybe immune to a certain level. I just want to remind people that the literature at this point has several articles that state that natural infection to coronavirus is not that robust. There's a range of immunity that you can get when you're infected from coronavirus. We don't necessarily know what level of which that is, that it's protective. We don't know how long that immunity lasts. What we do know is that it wanes fairly quickly. Those publications are coming out now. If you are banking on immunity being protective, so to speak, I would say that we're still kind of geared towards the vaccine being the better way to go for that.
And on a personal note, as someone who actually had COVID-19, got sick, completely recovered, I would much rather have had a vaccine than to be really sick for 12 days. So I do not recommend it.
I don't either. I do not recommend herd immunity by infection.
Yeah.
Okay. And the final question is very much about sort of how many people need to get this vaccine, right? Let's just say we project into the future, we actually have something that we know works. How many people need to get a dose of this vaccine, or however many doses it might require, in order for us to feel like, the population, we're safe, this thing is not spreading anymore?
There are models that say that 70% of the population needs to be immune before we can return to normal. So that's
okay. So
it's a lot.
Yeah.
All
right. And we see even in cases when and the thing is, until you have immunity, you don't have immunity. Like even in things like measles and other, where most people are vaccinated, you can quickly have outbreaks, right? So it's that same thing of like, it's okay, but you don't actually want it to be you, right? So that's the challenge that you have. You don't want to have.
So we're talking about billions of people, right? If we're talking from a worldwide basis,
billions.
Billions and billions of people.
Yes.
Yeah.
All right. Okay.
Which is quite a market for these vaccines, which is why everybody is working so hard on them.
Yeah.
Sure. I think that is all the time that we have. I actually wish that I could talk to you guys for far longer. I have still more questions. This is really interesting. And I just want to thank you all, Dr. Sabeti, Dr. Richardson, Dr. Corbett, for spending some time with us and actually really diving into these difficult questions.
Thanks a lot. And nice to meet you both.
I love all you ladies. So
I love this. We should love it.
yeah.
Absolutely.
This is really great. This
Yeah.
is really great.
Thanks.
Bye,
Thank you so much.
Bye.